All right, so as you can see from the title of this post, we are going to be doing a pre-lab today for the diffusion lab, which is kind of why I wanted you to do your conversions and calculation practice last week so that you could get your mindset in the right place for doing surface area to volume ratios and working with numbers. So we're going to take a look now at we're going to introduce the diffusion lab today. The homework is going to be for you to finish up the pre-lab questions that are associated in assignment post right after this one with what I'll be talking about. Chapter 4, Summary and Learn Smart are going to be assigned and due Sunday at midnight. So they're going to be your last grade before report cards come out. Test corrections. I want to make a point. So I can post test corrections or you can keep your test grades. If I post test corrections, it gives you a chance to earn points back. So every point that you missed, you can earn 70% of that back. Now, for some of y'all, that will make a difference between getting a passing grade or a failing grade. The problem is, not everybody has taken their exam yet. And I can't post this test until everybody takes their test. Otherwise, if I post this test, that means that people who haven't taken their test get an automatic 0 that they cannot change. So I just want to make that warning that I will be posting test corrections for next class so that people who want to do test corrections have a couple days to do them, but not that much time. But this also gives a chance to people who have not yet taken the test to go ahead and take the test so that you can, get a grade in for it instead of having a 0 on it, because that is 200 points and that's a lot. Okay, so our learning objective is that learners will be able to explain how cell size and shape affect the overall rate of nutrient intake and the rate of waste elimination. Now, learners will be able to make claims and predictions about natural phenomena based on scientific theories and models. So before we get started on actually moving into the pre-lab, I want you to pause and look up these definitions for these terms and write them in your own words in your journal, because these are important key vocabulary for you to know. So take a pause and write these definitions down. All right, moving on. So again, we've gone over this picture before. Cells range in size from 1 millimeter down to 1 micrometer. And cells need a large surface area of membrane to adequately exchange materials. So we've talked about this before when you did your calculations. And if you saw your comments that I made on your calculations that you turned in, when the cell gets really, really, really big, the amount of surface area compared to the volume actually is decreasing. So your surface area to volume ratio gets smaller and smaller as the cell gets bigger and bigger. Now that becomes a problem, because if there's not enough surface area for the cell to exchange materials, whether that be waste or nutrients, then it's not as efficient and the cell has a hard time maintaining homeostasis. It has a hard time getting rid of toxins and wastes, and therefore the cell cannot survive. So at that point of getting too big, a cell would much rather divide into 2 smaller cells than continue growing in size. Okay, so we're going to actually observe the efficiency of diffusion when it comes to cell size and surface area to volume ratio in a lab. So cell theory, just quickly, if you want to write this down, all organisms are composed of cells. So anything that's alive is made out of cells. All cells come from pre-existing cells. So if there was no other cell, then you can't get another life form coming out of that thing, right? Cells come from cells because cells are living. So they got to come from another living organism. And then cells are the smallest structural and functional unit of organisms. If you get smaller than a cell, it's no longer something that is alive. It is only something that's part of something that's alive, okay? So cells are the smallest structural and functional units of organisms, right? Small organisms we know are like bacteria, right? They're alive, but they're small because they're single celled. We went over these calculations and you did the 2 centimeter and the 3 centimeter ones on your own. And so the main takeaway that a lot of y'all got is that the larger the cell, the smaller this surface area to volume ratio gets. And then the less efficient that the cell is at absorbing nutrients and getting rid of waste. So like I had explained earlier, the bigger the cell is, the less efficient it is at doing exchanging processes. And when it's less efficient at doing these things, it has a harder time surviving because it can't get its nutrients to the whole inside of the cell easily because it can't absorb enough. And it can't get rid of the wastes fast enough. And so the waste stay in the cell for longer or more than it's supposed to. Okay, so that becomes an issue. And then a cell isn't going to continue growing anymore if its surface area to volume ratio is past a certain, is too small. We also did these calculations. So some of y'all did these correctly and some of y'all completely missed the point, which is okay because we're going to go over it, but it would have been nice if you had gotten it. So if you do this normally to do surface area, obviously you do each of these faces areas times 2 because there's one on the front and then there's one in the back here that you can't see. And then you do this face and this one, and this one, you add them all together and you get your surface area. And your volume is just the height times the base times the width, and then you divide your surface area by your volume and you get that ratio. So the ratio for this rectangular prism is a 1.04. Now, if you take that same exact volume, right? So our volume is not changing. It's still this times this times this, right? Because when you add all the little cubes up, it's still the same exact volume. The difference is that when you separate the cubes into 27 little cubes, that means every single cube has now 6 sides that it can absorb things on instead of one big face that it's absorbing things on. So this has 6, this has 6, this has 6. The ones in the middle each have 6 sides. The ones on the back each have 6 sides, okay? So that means that there's a lot more surface area now because you're going to take the surface area of each little cube. So 3 divided by 3 is 1 millimeter. It's 1 millimeter tall. 9.3 is 3.1 for the base, and then 12.6 divided by 3 is 4.2 as the width. And so when you multiply out the surface area for each small one, you then get to times a 27 factor because there are 27 cubes. That means the surface area is a little cube times 27, okay? And when you get a much bigger number, so therefore your surface area to volume ratio is a 3.12 instead of a 1.4. And you notice that this is actually 3 times bigger than 1.04. So when you have the same volume, but a different setup, you get a lot more surface area ratio. And this is exactly what happens if a cell that is really, really big divides itself into 27 little cells instead of staying as one big cell. So the ratio is much better. And then more things are absorbed and more things are exchanged out as waste. So these are the calculations written again out for you. And that's basically what this comparison is right here. So this is the smooth cell, right? The big rectangle. And this is the 27 little cubes version, right? So there's a lot more surface area, which means there's a lot more absorption and waste elimination occurring because on the same volume of the cell overall, there's a lot more surface area for the cell to interact with the outer environment, right? When the cell gets too big, the nucleus stays the same size. So you're trying to get materials from the outside all the way into the nucleus or from the nucleus waste all the way out. And it takes a lot longer because there's so much more volume and the surface area doesn't expand at the same rate when the cell is growing, okay? Over here, you can see it's got a little bit of distance to travel. So actually these exchanges happen really quickly. So the smaller the cell is, the more efficient it is at all of the exchanging processes, whether it's nutrients or wastes. Okay, so in order to go into the lab, you kind of want to get into the mindset of what is the most efficient surface area to volume ratio, right? You want to think of which surface area to volume ratio gave the fastest diffusion rate. Was it the bigger one or the smaller one? Which surface area to volume ratio had the greatest diffusion depth, right? Which one would diffuse in the fastest? That's what that question is asking. Smaller or larger? How might a cell's shape influence the rate of diffusion? That's when I want you to think about these two cells, right? A smooth surface or a surface that has divots and ridges in it. Which one would have a faster rate of diffusion? Which one would have a slower rate of diffusion, right? What factors affect the rate of diffusion and how can these be tested, right? Can you think of anything else? If not, just list the ones we've been talking about, okay? So I want you to write a formal if-then statement, or sorry, hypothesis about the relationship between cell size and rate of diffusion. So basically a format is gonna be like, if a cell has a smaller or larger size, then the rate of diffusion will be faster or slower. And you're going to tell me which combination of those you are going to predict is true inside of a lab setting. You can also predict one for a shape because we're going to be doing different size and different shape. So this is essentially what your actual lab paper was going to say. But since we're not printing it out, I have it on here instead for y'all. So you can pause to read it if you want, or you can just listen to me read it now. So for procedure, you're going to do a surface area and cell size comparison and diffusion rates, right? Diffusion, so basically absorbing materials into the cell. Because cell size and shape are important factors in determining the rate of diffusion, students begin their investigation of the movement of molecules across cell membranes by exploring the relationship between surface area and volume. Ask students to consider cell shapes, especially those involved with nutrient uptake, like in your intestines, Good examples include intestinal villi and root hair cells. Students should predict which measurement, surface area or volume, has the greater influence on the rate of diffusion. They should then calculate surface area to volume ratios and determine the diffusion depth and rate in agar. So we're going to do agar cubes, which is like this powder that you dissolve in water, and it becomes kind of like a gelatin, except agar is made out of a seaweed. So it's like a vegan gelatin, I guess. For data analysis, you'll answer these questions. You can think about them now, but you're going to actually answer them at the end in your report. So it's similar to the ones that you already talked about, but we're going to do them with direct data results. So again, which surface area to volume ratio gave the fastest diffusion rate? Which surface area to volume ratio had the greatest diffusion depth, right? How much diffused inwards? How might a cell's shape influence the rate of diffusion? And what factors affect the rate of diffusion? How can these be tested? Okay, so we will be using agar cubes that are very, very, very basic. So I have put NaOH, a very strong base, into these agar cubes. And I have dyed them with a bright pink dye known as phenolphthalein. Okay, we're going to be putting them into an acidic situation, which is hydrochloric acid, which is HCl. And we're going to be observing how fast does the HCl diffuse into these agar cubes, right? So the phenolphthalein is bright pink when it's in a base, and it turns white when it gets neutralized by the HCl or turns acidic. Okay, so we're going to be essentially watching these bright pink cubes slowly turn white from the outside to the inside. And we're going to be calculating how deep the white goes and how fast the white goes inwards from the outside. We're also going to be calculating the volume that is turning white and the volume that remains pink. Okay, so it's going to be a little bit tricky, but I will explain it. Okay, so in this, make sure that when I do the demo, I'll be wearing gloves and goggles to make sure that, again, I'm being safe because I'm using strong acids and strong bases. Okay. Lab safety first. So again, like I said, phenolphthalein, when it's really, really basic, like when I put in a lot of NaOH, it's going to be this bright pink fuchsia color. Once it gets neutralized and it's below a pH of 8, which is still kind of basic, it's going to be colorless or white. Okay, so it's going to return to the original agar cube color, which is white. So when a base and an acid touch, they neutralize and they go to 7, but it's going to be sitting in a acid solution, so it's going to be more acidic, but anything underneath or more acidic than 8.2 is all just going to be colorless or white. Okay, so again, remember, acidic is anything from 1 through 6 on the pH scale, 7 is neutral, and then 8, 9, 10, 11, 12, 13, 14 are all basic. Okay, we're going to be starting around like 12 because it's super, super basic in the cube, so we should see fuchsia from the start. But then as we go on and as the HCl or hydrochloric acid diffuses inwards, parts of the cubes are going to start becoming colorless. Okay, so before I go on to that, I want to have you guys do a little calculation for me. So notes on making solutions. I actually made these solutions for y'all already because I made the agar and the HCl solution is pre-made as well. So notes on making solutions. We usually go by percent, right? So I made a 2% agar with phenolphthalein. That means 2% of the solution of the agar is actually the agar powder, and the rest is a liquid that it is dissolved in. So percent symbol or percent is out of 100 or divided by 100. So this actually represents grams of your substance divided by 100 milliliters of a solvent. So in our case, that 100 milliliters is water. Okay, and the grams we're looking at is how many grams of agar we're putting into this 100 milliliters of water. Right? So for example, if you wanted to make a 2% salt solution, that would mean you need 2 grams of salt put into 100 milliliters of water. And that would be your 2% salt solution. Okay. A 1% salt solution is 1 gram divided in 100 milliliters of a solvent. Okay. So you could put salt, you could put HCl, it comes in a powder form too. You could put in sugar, right? If you put 1 gram of this into 100 milliliters of liquid, then you have a 1% salt solution or you have a 1% sugar solution, okay? Now, I want you to calculate what you need for a 750 milliliter amount of a 2% agar solution. So remember, 2% means that there are 2 grams of agar in 100 milliliters of water. But I want 750 milliliters. So on your paper in the assignment in the following document post, you're going to want to do this calculation for me. And how do I get 750 mils of 2% agar solution. How much agar do I need? How many grams? Okay. And I will give you a hint for your setup. Okay. So if you have 2 grams over 100 mils, that's the correct concentration that you want. Now, what you're trying to find out is how many grams of agar you need to add into a 750 milliliter amount of water in order to get the right concentration, right? So you're going to be cross multiplying and then solving for this question mark. Okay, and I will be grading that. So essentially you're going to be starting out, like I said, with a fuchsia agar cube, and we're going to have 2 sizes. And then they're going to be put into a container with HCl, and they'll slowly turn white from the edge going inwards, as you can see, as the HCl diffuses in and neutralizes the NaOH inside of the agar. And then the phenolphthalein the color will recede as it's being neutralized. So what is the purpose of the lab? You're going to write me a hypothesis, right? You already wrote it earlier. You're talking to me about if a larger or smaller size has a faster or slower diffusion rate based on, the size, right? You're going to be talking about what variable we're changing. What is the independent variable, right? It's related to this picture here. What variable are we measuring, right? I said earlier, we're gonna be measuring something related to the white part appearing and growing. How will you measure that dependent variable? Okay, are you measuring volume? Are you measuring area? Are you measuring distance? You tell me. And you want to tell me what kind of measurements you're going to be using, right? If it's only going a little bit at a little bit, are we going to be using meters? Are we going to be using centimeters? Are we going to be using millimeters? Okay. What conditions are we holding constant? Think about the conditions in the room that we want to hold constant so they don't affect the results of the experiment. We're going to make a table that I'll show you on the next slide on how to record your data. And you're going to actually make 3 of them, okay? And they're going to have different titles. And then you're going to try to think about what you're going to graph at the end. Which surface area to volume ratio had the greatest diffusion depth, right? Which did the biggest cube, the smallest cube, or the different shape have the greatest diffusion depth? You're going to try to tell me also which gave the fastest diffusion rate. Was it the big cube, the small cube, or the different shape? These are all just predictions for now. It's okay if you're wrong. Okay, so basically you're going to be making a data table like this. You want to have a column for time, a column for distance of white, right? This is going to be your diffusion depth. You're going to have a volume of pink that you're going to calculate. And then you're going to have a volume of white that you're going to calculate. Okay, so we're going to be calculating how much pink is left on the center of your cube. We're also going to calculate how much volume of white has been appearing as the HCl diffuses from the outside to the inside of the cube. And you're going to want to leave the bottom of your page for calculations because you're going to be doing calculations. It's not really going to be every 15 minutes. These are going to be an example, but you want to leave a box for calculations. Okay, you're probably going to be doing as many as you have here. And we might be even doing them 30 or 45 minutes apart, depending on how fast diffusion occurs. You're going to want to make, like it says right here, you'll need 3 tables because we're going to be doing, let me show you. We're going to be doing 1 table for the big cube, 1 table for the small cube, and 1 table for a different rectangular prism, so a different shape. So you're going to want 3 pages or 3 data tables. Okay, and then I'm going to have you lastly drop off by thinking about this problem. So how do you calculate volume for the green cube on the inside of the blue cube, right? I showed you that the blue has diffused in by 1 millimeter on all sides, right? So you know the original blue measurements was 9 millimeter base, 7 millimeter height, and 4 millimeter depth. But I want you to calculate the green volume now, right? So for example, you know that if this went in for the base, it went in on this side by 1 millimeter. You also know that it went in on this side by 1 millimeter. So you're technically going to be having a base of your green be 9 minus 2 because there's a 1 millimeter difference on the left side and a 1 millimeter difference on the right side. So your green base is going to be, 9 millimeters, minus 1, which is 7 millimeters. Over here, you have your 7 height, same idea. You're going to have 1 millimeter taken off the top and bottom. So you're going to have your 7 millimeters minus 1, and that will be 5 as your height. Okay. And then lastly, you have your width. You have 1 missing from this side and 1 millimeter missing from this side. So you're going to be 4 minus 1, which is going to be 2 millimeters. And so basically, after you've done all of those subtractions, you should be able to find the inside or the green cube volume just by doing your new base times height times width, which is 70 millimeters cubed. Now, how would you calculate the volume that remains blue? Okay, remember the whole inside is green now, which means we're not looking for the inside volume. And you can't just calculate 9 times 7 times 4 because that would be the whole volume, right? If there was no green on the inside, then calculating the whole volume would be the blue volume, but we're trying to exclude the green, okay? So here's where it gets a little tricky. You're gonna do your original blue volume, right? So base times width times height, which is 9 times 4 times 7, right? So you get your volume of your blue overall cube. But we need to eliminate or subtract out the green part in order to just get this blue outline. So if like you hollowed out the green part of the inside and you had an empty blue shell, that's what we would be talking about right now. That's the volume we want, okay? So basically you'd subtract it. So you would subtract your volume of your blue from your volume of your green to get the volume of blue remaining, right? That blue outside shell. And you just substitute in the two numbers that you already found from doing your volume of your green and your volume of your blue. And then you end up with 182 millimeters cubed, which is the volume of the blue outside left. Okay, logically, if this doesn't make sense, definitely ask me about it because you're going to be doing these two sets of calculations for every single data point that you collect throughout this lab. And there's gonna be 3 data tables. Luckily for you, we're only collecting a few data points because it does take a long time for these labs to process, okay? But again, I'll be collecting that data for you. So again, if you have questions, make sure you ask. That's all for today. After you finish up your pre-lab questions, to the best of your ability, turn them in so that I can grade them.